Well, hello everybody. So today we'll be talking about highly repetitive ribosomal RNA gene-containing regions, also known as Nucleolus organizer regions, and I will show how we study these regions using Oxford Nanopore sequencing technology and what we learned so far. So in Arabidopsis, Nucleolus organizer regions are located at the tips of chromosome 2 and chromosome 4. These regions span across several megabases and contain hundreds of tandemly repeated ribosomal RNA gene units. Each unit is about 10 kilobases. Ribosomal RNA genes are very similar to one another and cannot be discriminated using short-read sequencing approaches. So we took advantage of ultra-long sequencing reads, which span across several ribosomal RNA gene units, but also they were accurate enough to discriminate large structural variations, such as insertions and deletions, sometimes found within individual ribosomal RNA gene units. So with that, we were able to assemble both Chr2 and Chr4 regions in Arabidopsis. Chr2 is about 5.42 megabases and contains 518 ribosomal RNA gene repeats. Chr4 is a bit shorter. It is 3.85 megabases with 372 ribosomal RNA gene repeats. And this identity heat map for both Chr2 and Chr4 generated using stained glass tool. And you could see that in both Chr2 and Chr4 there are some regions, which is here colored in dark red, with very high identity. It contains ribosomal RNA genes with identity approaching 100%. So this suggests that ribosomal RNA genes in Arabidopsis do not evolve independently, but likely undergo concerted evolution. To study distribution of active and silent ribosomal RNA genes within NORS, we first looked at cytosine methylation. And these cytosine methylation profiles for both Chr2 and Chr4. And in plants, we have three different cytosine methylation contexts, CG, CHG, and CHH, where H is any nucleotide but G. So if you look and compare Chr2 and Chr4 methylation profiles, you could see a striking difference. If you look at the first track, CG methylation profile, you could see that Chr2 is predominantly heavily methylated. In contrast, Chr4 has both high and low methylation profiles. The central part of Chr4 is low methylated, and this low methylated region spans across almost 2 megabases. But this low methylated region flanked by highly methylated region on both ends of Chr4. We also overlaid gene activity track at the bottom. And this track shows positions of location-specific single nucleotide polymorphism found within ribosomal RNA genes. Gray represents no polymorphism found, and it is basically a reference sequence. And vertical lines represent positions of those polymorphisms. Then we map expression, ribosomal RNA gene expression from RNA-seq data to the track, and we observed that most of the ribosomal RNA gene expression is coming from a low methylated region of Chr4, those green markers. So next we looked at a high order organization of NOR regions within Arabidopsis thaliana genome, and also we were interested to look at epigenetic profiles of NORs. To study high order organization, we took advantage of Oxford Nanopore Pore-C assay. And I'm not going to describe in detail this assay, but I just want to mention we tested different restriction enzymes. For today purposes, I'm going to basically show data with XBA1 restriction enzyme. It cuts Arabidopsis thaliana genome into average size of 2 to 3 kilobases. But importantly, it has one site per ribosomal RNA gene unit that is within ribosomal 18S ribosomal RNA gene. Another method which we are using currently, it is DNaseI assay. It stands for direct methylation and long-read sequencing assay. It is a very good assay to study epigenetic profiles of long, highly repetitive regions because it provides single molecule resolution for mapping protein DNA interactions genome-wide. It relies on targeting protein A fused HER5, which is adenine methyltransferase, to loci of interest via antibodies, and once it's activated, it can methylate adenines in its proximity. And then you can detect M6A methylation with Oxford Nanopore sequencing. So this genome contact map for Arabidopsis thaliana, which we obtained using Pore-C assay. So the main diagonal line which you see here, it is local interactions detected, and the signal of the main diagonal line will be long-range interactions. I don't have time, but I want to mention that Pore-C did capture all the known interactions within Arabidopsis thaliana genome. I'm listing below. But also identified some novel interactions as well. And specifically, we were interested in interactions of no regions with the rest of the genome. Surprisingly, we didn't identify any interactions of no regions, both NOR2 and NOR4, with the rest of Arabidopsis thaliana genome. Those regions actually form their own domains within short arms of chromosome 2 and chromosome 4. The only interactions we identified is between NOR interactions. There are points of interaction between NOR2 and NOR4 which we are interested in pursuing further. So in this slide, I'm further zooming in into each NOR. On the top is, again, Pore-C data, which shows more detailed information about organization of each NOR. And you could see here that both NORs have additional subdomains. NOR2 has several subdomains. NOR4 has two major subdomains. Below, I also put some CG methylation profiles, which I showed you earlier, as well as additional tracks, like chromatin accessibility track, and also H3K4 methyl 3 active histone modification track, which we obtained from DNaseI experiments. And from those epigenetic profiles, you could see how strikingly different both NORs. For example, consistently with low CG methylation within central part of NOR4, you could see that the same region actually very accessible, kind of open, present in an open chromatin conformation. The same region also has active histone modification mark H3K4 methyl 3. So zooming in further, here I'm showing representative 10 ribosomal RNA genes from NOR2 and central region of NOR4. You could see again how these two regions are strikingly different in respect of CG methylation, DNA accessibility, and H3K4 methyl 3 modification marks. And it is, and now the same zooming in further to individual ribosomal RNA gene from NOR2 and NOR4. You could see through the whole body of ribosomal RNA gene within NOR2, you could see that it is highly methylated, very inaccessible, and also has very low H3K4 methylation mark. In contrast, NOR4 has completely opposite profiles for all the three markers. So in conclusion, I would like just to say how important it was to be able to assemble both NOR regions within Arabidopsis, because it opened us opportunities to study activity of individual ribosomal RNA genes and also three-dimensional organization of NOR regions. We found that NORs do not interact with the rest of the genome, but possibly interact with each other. But we want to explore this further. We also observed how strikingly different both NORs are in respect of epigenetic profiles, with NOR2 being more highly methylated and more condensed chromatin states, whereas NOR4, the central region, likely to be expressed. So I would like to thank Professor Craig Pigard for his guidance and support, and also I would like to mention that we have a very great team, and it's very nice to work with. Especially would like to thank Dallin, Ramia, and Wei, postdocs in Craig Pigard's lab, who contributed to this project. And also I would like to thank Professor Nicholas Artimos. He was very helpful to establish amino sequence in our lab, and also very helpful to with bioinformatics pipelines. Thank you.